to record.
Can we start, Miss?
Okay, sure, please.
Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, everyone.
Good morning.
Good morning.
Morning. Okay. Today, class, we're gonna learn about the engineering in animal, right? So our presenter today is Muhammad Fahriza Amri, and we would like to greet him. Okay. Hello, Fahriza. Are you ready?
Hello.
Are you
Yeah, I'm very much ready.
Okay. So
All
the times is yours.
right. Thank you very much, Alicia. All right. So let me share my screen. So here is my, hurriedly put together my mind map. So let's get started. So we're going to talk about gene engineering in animals. So we have definitions, types, process, and products. Let's start with definitions, shall we? So as you can see, the definition of gene engineering in animals is that change of animal DNA that does not occur naturally. As you all know, when animals, sexually breed with each other, they pass along traits from their parents. That's why they are, a mix of what their parents are. But the thing is, when they do that, the traits that are going to be inherited are pretty much random. You might get a breed of animals that is just the perfect version of what you need, but at the same time, you have the equal chance to get animals that have traits that you don't desire. And thus, there comes gene engineering, especially in animals. What it does is that it basically, make sure that the offspring that produce express trait that is desirable to what you want that animal to express, what traits that the animal you want to express. So, all right. After we talk about the definition, let's move on to the types. So we have 3 types of gene engineering, which is, as mentioned on the last meeting, which is DNA recombination, gene transfers, and gene editing. So to sum it up, DNA recombination is when you put together genetic materials from 2 different organisms so that the end result would be the desired trait of, let's say, that 2 organisms. So what you do is that, let's say you pick the gene from this animal, and then you put also from that animal, and then you put it into a vector, which is probably going to be a virus, and then you put them together, you inject it to your desired animals, and then that animal will start producing the traits that is inside of that gene materials. And then you have gene transfer, which is a more direct way of DNA recombination. Instead of combining DNA, you basically just select traits that specifically will express this trait. You just genetic material that is exactly going to produce this trait, and then using the same method, which is introducing it with a virus, and then suddenly you have the animal who produce traits that you wanted. For example, in a cow, that it produces, let's say, more milk or better meat, or is immune to certain diseases. That's what you do. And then you also have the last one, which is gene editing. This one's a bit different because this one, you do it as the animal undergo the embryo stage. So what you do is that you take let's say, the egg cells and the sperm cells, and then what you do is that you take out their genes, you take out their chromosomes, you either turn off genes that can cause defective traits, and you turn up genes that are beneficial to the animal. And then you insert back those chromosomes, and then you let the animal, grow into genetically engineered animals. And then let's move on to the process after we talk about the types. So the first process, the first step, we all know it's going to be to identify the trait that we want. For example, again, as mentioned before, do you want animals that produce better milk or better meat or is resistant to disease? We see in the wild if there's any animals that express this trait. And then the step two is to identify the genes that produce this trait in that animal. And after that, connect it to the host. So that would be the process of gene engineering in animals. And then we go to the product, which is, well, you could say traits that are expressed for commodity value, which is here is an example of it, which is glowfish. It's a fish that express enzymes that are bioluminescent so that they can glow in the dark. And then you have also animals who, as mentioned before, has livestock value, moral livestock value. And yeah, from that, as you can know, that gene engineering in animals sounds very amazing because it produces things that supposedly are superior to the natural ones. But then again, there are some concerns. First of all, there are side effects in individual animals because again, while we so that they produce desired, let's say desired products, desired traits from that gene that we introduce, the thing is, we don't know how those genetic materials will interact with the body of the animal that we are editing. We don't know, like, for example, if we edit their genetics to produce a certain protein, will that interact with the other proteins that are already inside of the animals and then like cause harm to the animals? We don't know that. So that's one of the concerns. And then the other one is that the effects to the environment. This is a more wide consequences of the previous ones, which is that again, what if we introduce, the genetically engineered animals to the wild? What would happen? What if they interbreed? Would they replace the wild type of animals and then making that, wild type of the animals extinct? Or would they produce offsprings that are defective, but also, produce really fast that they replaces all the original that is supposedly better? Or that, they might become an invasive species which dis- destroy the environment and the ecosystem around them. And then you also have the large amount of resources required because, as much as gene engineering is an amazing technology, the cost to do that is quite a lot because as much as you like to introduce traits to animals, most of the time they do not survive the process. So that, let's say you have 100 offsprings that you want to gene engineering. Most of the time, there's only like less than 10 that survive the process and becomes the full animal that is genetically engineered. So yes, it requires a lot of resources and it has a very big fail rate. So there's that. So yeah, that's all from the genetic engineering in animals. If you guys have any question, go ahead and ask.
All right, thank you, Fahriza, for the presentation about engineering in animals. So, for those of you who have a question, please kindly raise your hand, Diki. And then, no one? Dela, Miss Lilith, two more. Okay, maybe while we waiting for another friend to raise their hand, we begin with Diki.
Go ahead, Iki.
All right, thank you very much, Fahriza, and thank you very much for the presentation, Fahriza. So my question is, you said that something interesting, like what will happen if you're gonna, take this genetic engineering, the genetic engineered animals to the wild? What do I think about is, that our world is gonna be different, like the past and the future. Maybe we can just, making the gene much more durable to the condition of the future itself. Like we are talking about other stuff like what the world itself, like global warming and other stuff, because there's a lot of factory and the animals came out from the forest going to the village or to the town because the forest is not stable anymore. So can we do, with the genetic engineering there, like making the animals more durable to the condition of the future? That's my question.
I see. Okay, thank you very much, Iki. Yeah, I would like to keep the questions. I would like to go on to the next person to ask.
Okay, let's please Della. Question from Della.
All right. So first of all, I want to say Fahriza has a good presentation. So I wanna ask, can animal do crossbreeding? I mean, in different types of animal, for example, lion and tiger. So how it can happen? The reason.
Okay, thank you Della for the question. Next, maybe Miss Lilith.
Okay, next for me, Miss Fahriza.
Yes,
Yes.
please.
Okay, thank you. Thank you very much, Fahriza, for the great presentation about the gene or genetic engineering in animals. I would like to clarify about the types in here. The number one is DNA recombinations as the types of the genetic engineering in animals. I would like to clarify, is DNA recombinant is same or different with the DNA recombinant technology with Miss Arni presentation in last week? Okay, is
Okay.
same or different? And then is same or is different or same? What's the relationship between, is there any relationship between DNA recombinations technology with genetic engineering in animals? Because this is focused on gene or DNA.
Yes.
Okay, thank you.
Okay, thank you very much. Yes.
And so the first question is from Dicky. If I summarize question from Dicky, it will be, can we make our animal more durable for the better future? Is it right, Dicky?
Well, yes, simply say it's like that.
Maybe Fahriza need time.
No, I actually would like to answer now. So regarding that question, whether we can make our animals more durable to, let's say, climate change or let's say another pandemic whatsoever, yes, we can actually do that. But then again, just introducing the traits that make animals more durable to disease and everything, it requires first of all to find the animals in the wild that are actually are adapted to, let's say, global warming. So basically we have to find the animals first, and then we have to see how, let's say, the genetics of that animals are expressed, to see the traits are expressed, because again, we don't want to introduce let's say traits that make them resistant, but then at the same time ruins, basically making, let's say, the meat of the animals that we are making as livestock to be inedible. You don't mind, like, you don't want to have animals to become resistant to heat, but at the same time they turn into a lizard, for example. That would just be disastrous. So yeah, you can, but then there's a lot of consideration before you do that.
Yeah, so the answer is you can. How about Vicky? Are you satisfied with the answer?
Well, yeah, I just think the same too, because it's just a crazy idea itself. Like I could, thinking about taking a genetic from the cacti and going to make it to the forest plants, and so the forest plant doesn't require more water or something. I mean, it's crazy idea, but maybe it can work, something like that. Thank you for.
Yeah, thank you. Another question from Della is, can animal do crossbreeding, for example, from another species to another species? And if we can, what is the reason? Do you need time?
No, so I would like to answer that. So if you're talking crossbreeding in a traditional sense, as in, just have the animals breed together, it's pretty much impossible because again, the germ cells, or the cells that is like the eggs and sperm cells has in each animals, let's say it has different receptors. So you can't just have, let's say chicken sperm, breed with, let's say, duck eggs and create something new. You can't just do that because again, they are very different. As in, combining the DNA artificially, as in, you do it in the lab, as in again, uh, using, let's say, DNA recombination. Then yeah, you can. There's several animals who are, created that way. If I remember, like some breeds of dogs are created that way. And then there's also, few livestock animals that I don't really remember, but yeah, they are made that way. So yeah, it's possible, but then you cannot just do it, naturally.
Okay, so from Fahriza, we can conclude that it is impossible because the cell of one species to another is very different. But for artificially, it is possible. What about Della? Is it?
I got the answer. Thank you, Fahriza.
Okay.
And the last question is from Miss Lilith. If I'm not mistaken, the question was, what is the difference between gene engineering in animal and DNA recombination? Is it true, Miss?
Yeah, I focus on types in here, DNA recombinations, because in last week we have already learned about DNA recombination technology with Miss Ani. So my question is it same or different with DNA recombination in here in Fahriza's presentations? Or is there any relationship between DNA recombination and genetic engineering in animals?
So as Miss Lilith asked in the term of the types. How about Fahriza? Do you need time?
No, actually, I would like to answer now. So regarding that, DNA recombination and DNA recombination in technology is by concept the same, but then by meaning it's different because DNA recombination itself is like the process, while DNA recombination technology is more like what do you use to facilitate that process. So here I'm talking more about like what happens there, and on the other one, it's more discussing more about like how do you do this. So that would be the difference. But again, like in the basics, they are pretty much the same. They are both DNA recombination. It's just that what it discusses is very much slightly different, so to say. And for the other question, which is can you use DNA? What is it? DNA recombination technology in gene engineering in animals? I mean, yes, of course you can use it because it is, again, as I mentioned before, it's a technique that you use to apply, let's say, the genetic engineering in animals. It's one of the techniques that you can use, and so yes, it is very much related to the both of them are very much related to each other. That will be my answer.
Okay, thank you, Fahriza. How about Miss Lilith, Miss?
Yeah, thank you, Fahriza. So maybe we can sum up. So DNA recombination technology in Arni's presentation in last week as the basic for making genetic engineering in animals. Can we say?
In a way, yeah.
Okay. So DNA recombination technology as the concept or the process. So the implementation we can see in genetic engineering animals and maybe on the genetic engineering in other living things, right, Fahriza?
Yes.
Okay, thank you, Fahriza. Thank you,
Okay.
Alisa.
Okay, thank you. Because there is still time, so anyone who have a question, please raise your hand. Or okay, Anthony.
Okay, so my question is among the types that Fahriza mentioned before, which one do you think that can be better to do? Is it my voice clear?
Yes. So Anthony asks in the term of types, which one do you think it's better to do? Fahriza, do you have
Yes.
time?
Thank you.
I can answer now. So that depends. I mean, do you mean better as in like better resulting, or that you mean better, let's say, outcome? Because those are different. So basically, to have better result, better animals that came out of it, I would suggest you probably use, let's say, DNA recombination, because you can introduce much more traits into the animals directly. If you want to be more accurate with the traits that you want to introduce, it would be gene transfer. But then if you want to be safe, because you're not actually doing much to the genetic material of the animals, you can use gene editing, because again, the gene materials is pretty much still the same. It's just that you turn off this and then you turn on that. It's basically just that. So yeah. As for the better ones, it depends on like what do you define as better.
Okay, that's the question from Fahriza. And what about Andini?
Okay, thank you very much, Fahriza, for answering.
One more question from Hesti.
Thank you, Felicia. I want to ask Fahriza, if the genetic engineering is successful and then it will produce new individuals, right? So, is it possible that that new individual will, for example, they have a disease and then it will lead new disease? Is it possible? That's all.
So that's the question from Hesti. If genetic engineering is successful, is it possible if an individual got the disease? Is it true, Hesti?
Yeah, is it possible lead new apa ada penyakit baru gitu? For example, there is avian flu, right?
Yes.
Is it possible? It's the disease come from the new individual.
I see. I mean, yeah, that's a good question. So I would like to answer that. As far as I'm aware of, I don't think it can produce new diseases, because again, the concept of diseases, especially viruses, is that they need to be able to infect first before they can evolve. So basically, let's say you have animals that are resistant to a disease. So there's basically just little chance for it to like produce new viruses, because again, they don't get infected in the first place, so that the virus cannot like mutate and adapt to them. So I think it's unlikely. I mean, nature's find a way, so it's possible, but it's unlikely.
Okay, thank you, Fahriza, for the answer. How about Hesti? Is it satisfied?
Yeah, okay, I got it. Thank you, Fahriza.
Well, I think the time is up. I would like to appreciate Fahriza first for great explanation about animal engineering. Fahriza have explained the content very well in an engaging way. Engaging way, but I would like to summarize the point that Fahriza has explained. The first is Fahriza explained about the definition, which is the changes of DNA that doesn't occur naturally. And he also explained about the types, which is there is 3 types. Also, Fahriza explained about the process. And the last, also the example. And the last about we have to be like careful, because of course it has the side effect, whether it's to individual animal or environment animal. So I think that's all for this session. I would like to close the first session. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.